A big hello to all my future doctors out there. So today I am here with the 7th day video, right? Today is the final day that we have, and I have chosen just one chapter, and that is biomolecules. Why, ma'am? Why have you chosen just one? Because this chapter is one of the high weightage chapter of your 11th grade. Actually, it carries the most amount of marks. That is basically 6 questions are gonna come. 6 into 4, 24 marks you can get from this particular chapter, which is why I thought I should be making a separate video on it, and I hope you enjoy it. If you enjoy it, do let me know in the comment section as well. And my dear children, you should not be waiting for Jasmine ma'am to tell the answers. You should be solving it by yourself as well. So whenever you see a question, pause the video, try to solve it, and then check whether your answer, my answer is the same or not. Works for you. If it works for you, it works for me as well. Chalo, let's begin. There are 40 questions. I hope we are ready to do it. So identify the basic amino acid from the following. You have tyrosine, you have glutamic acid, you have lysine, you have valine. Starting from valine. Okay, valine basically is a neutral amino acid, neither basic nor acidic. It will be what? Neutral. On what basis do we say it's acidic or basic, my dear children? You know, an amino acid is made up of what? An amino acid would be having, okay, an amino acid will be having an amino group, would be having a carboxyl group, would be having an H, okay, and would be having an R group right over here. And this R group, whatever is there over here, decides whether this amino acid which is there would be basic or say acidic or basically neutral. So valine structure, basically if you see, you understand that it would be what? Neutral. Talking about lysine which is there, lysine is actually basic in nature. Why? Because over here in the R chain right over here, it has an amino group which makes this whole structure what? Basic structure. Talking about glutamic acid, again it will be having a carboxyl group in its R chain. In its R chain, it will be having a carboxyl group which makes it acidic. Talking about tyrosine, tryptophan, phenylalanine, they are all aromatic amino acid. That means they would be having a ring shaped structure and things like that. Okay, you are asked about the basic amino acids. Your answer should be lysine. Lysine looks like this. So I told you it will be having an amino group. So amino group is this, and this contributes apart from the normal amino group which is there. In the R chain also there is an amino group which contributes it to be what? Basic in nature. Works? Okay. So it can become NH3 and things like that. Fine. Now, a typical fat molecule is made up of, a glycerol, okay, a glycerol and 3 fatty acids. To a glycerol molecule, 3 fatty acids can come and join, my dear children. You get it? 1 glycerol and 3 fatty acid molecule is the thing. So typical fat molecule is made up of 1 glycerol and 3 fatty acid. It is known as triglyceride. If you have 2 fatty acid, it is known as diglyceride. Diglyceride. Okay? Fine, this is how it works. So, 1 glycerol and 1 fatty acid molecule would be a wrong answer. That is not how a typical fat molecule looks, right? 3 glycerol and 3 fatty acid molecule, no. 3 glycerol molecule and 1 fatty acid molecule, no. 1 glycerol and 3 fatty acid molecule, yes. There will be 1 glycerol and attached to it will be 3 fatty acid molecule. Cool? Yes. So, this is how it looks. Glycerol and 3 fatty acid chains that you have. Now, going ahead, uridine present only in RNA is what? Is a nucleoside, is a nucleotide, purine or pyrimidine. Uridine which is there, my dear children, it's basically when uracil, okay, uracil when it binds with ribose sugar. Uracil is what? Uracil is what? A nitrogen base, yeah, that you only find in RNA. And now this nitrogen base is going to attach on the first carbon right over here. Nitrogen base is going to attach on the first carbon of what sugar, my dear children? Of ribose sugar, not deoxyribose. This is what? Ribose sugar. Ribose sugar is a part of RNA. RNA molecule is made up of ribose sugar and stuff like that. Okay, so uridine present only in RNA is what? A nucleoside, nucleotide, purine or pyrimidine. If the question was uracil present in RNA is what? It would be pyrimidine, because uracil is a pyrimidine. In nitrogenous bases you have 2 types, purines and pyrimidines, okay? And under pyrimidines comes uracil. You're talking about uridine. Uridine is basically known as a nucleoside. What do you mean by nucleoside? Basically when uridine bonds with the sugar plus the nitrogen base is known as which sugar? Uracil sugar. Sorry. Uracil, the nitrogen base plus the ribose sugar, together it forms uridine. Together this particular structure, whenever there is a nitrogen base, whenever a sugar is there, you call this structure as nucleoside. Suppose on the 5th carbon you have phosphate group attached, you will call this whole structure now nucleotide. nucleotide. This is nucleoside. The whole structure is nucleotide. You are asked about uridine. Uridine present only in RNA would be what? A nucleoside. Not a tide or not a pyrimidine, not a purine, on things like that. I might have marked pyrimidine, but the answer is nucleoside. Cool? Yes. Sorry for that confusion of marking it wrong. Yes. Now, which one of the following biomolecules is correctly categorized? Let's see. Characterized. Okay. Lecithin, a phosphorylated glyceride found in the cell membrane. Is it? Is it a phosphorylated glyceride that is found in the cell membrane? Let's keep it on hold. Palmitic acid, an unsaturated fatty acid with 18 carbon atoms. First of all, palmitic acid just has 16 carbon atoms, okay? And in that carbon atom, one carbon atom belongs to the carboxyl acid, no carboxyl group as well. Okay, so total 16 carbon atoms are there, not 18. Adenylic acid, adenosine with a glucose phosphate molecule. Wrong. Adenylic acid means adenine is there, that is nitrogen base, and then the sugar and then the phosphate. Together you call this whole nucleotide as adenylic acid. So adenosine with glucose phosphate molecule, no, there is no glucose phosphate molecule. Adenosine with phosphate molecule would have been still considered. This particular thing would be wrong. Now alanine amino acid contains an amino group and an acidic group anywhere in the molecule. No, it does not. Okay, it would not be containing any groups like that. So your answer is what? This only. Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. Okay, they have phosphorus or probably phosphorylated organic compound which is there. Okay, example of this is lecithin, and lecithin is a part of your cell membrane. Now palmitic acid has 16 carbons including what? Carboxyl atom, carboxyl carbon, I already told you. Now if phosphate group is also found in esterified, is also found esterified to the sugar, they are known as nucleotide. I told you sugar, nitrogen base is nucleoside. Phosphate group is there, nucleotide. Okay, adenylic acid, thymidic acid, guanylic acid, uridylic acid and cytidyllic acid are nucleotides. So whenever you have phosphate group, you call all of them as acid. Okay, now the R group in these proteinaceous amino acid, that is alanine. Okay, in this proteinaceous amino acid could be hydrogen, that is in the case of glycine, or probably could be methyl group, such as in the case of alanine. So in the case of alanine, it has just a methyl group, which is why it is what is not considered to be basic or acidic. It will be neutral. It will be neutral. So R group can be different. Please remember this also. Okay, it is just a hydrogen. This is the simplest amino acid, just as having a hydrogen in the R group, that is glycine. Would be having methyl group in the case of alanine, and in the case of serine, it will be having hydroxy methyl group. So the answer for this question is just A. Okay, cool. Next, lipids are insoluble in water because lipid molecules are. They are insoluble in water. That means they hate water. And what do you call this? Whenever you hate water, whenever you have a fear of something, what do you call? You have phobic to it, or basically you say that you have a phobia, right? Similarly, when the lipid molecule do not like to go in the water, what do you call this? Hydrophobic nature. So lipids are insoluble in water because lipid molecules are what? Hydrophobic in nature, not hydrophilic. Hydrophilic means water loving, not neutral, not zwitterion. Okay, this is just hydrophobic. Lipid is hydrophobic, which is why it is insoluble in water. Now, next question. The 4 elements that make up 99% of all elements found in living system are CH, carbon, hydrogen, oxygen and phosphorus. Carbon, nitrogen, oxygen, phosphorus, hydrogen, oxygen, carbon and nitrogen, carbon, hydrogen, oxygen and phosphorus, sulfur. What is the answer for this question? Your answer is straightforward. It would be hydrogen, oxygen, carbon and nitrogen. That makes up 99% of elements of our human body or living system, my dear children. Always remember, nitrogen is a part of a lot of things. You take DNA, it will be there. You take proteins, it will be there. You take fats, it might be there. Okay, but yes, so this is your answer. Next, secondary metabolites such as nicotine, strychnine and caffeine are produced by plants for what? For their nutritive value, for their growth response, for the defense action, effect on reproduction. What do you think? Why do you think they produce all this nicotine and stuff like that? Basically, all the secondary metabolites that have been mentioned over here, It's for protection against the predators that feed on them, my dear children. So it is going to act like a defense action. It is going to act like what? A defense action. Secondary metabolites such as nicotine, strychnine and caffeine are produced by plants for defense action. Works? Okay. Next, concanavalin A, I always tend to mess up the name, okay. Concanavalin A is what? A pigment, an alkaloid, an essential oil, a lectin. What is the answer for this question? And I'm telling you, the table of all the secondary metabolites are really important. Make sure you're going through it. Okay? Do you have any idea what is it exactly? So it is a lectin, my dear children. Lectin is something that would be having carbohydrate attached to it. Okay, it's a type of protein where basically it has a carbohydrate attached to it. So concanavalin A is a lectin and it is a secondary metabolite. It's not a pigment, it's not an essential oil, it's not an alkaloid, it is just a lectin. Next, which of the following glucose transporters is insulin dependent? That means, what do you mean by insulin dependent? For example, there is a cell. What happened? There are receptors on the surface of cell. Now on this receptor, what will come and attach? Insulin comes and attaches. When insulin comes and attaches, there might be certain transporter molecules up over here in the cytoplasm. Okay, what will happen when the insulin comes and attach over here? These transporter molecules would be going up, getting integrated right over here and would be letting in the transport of glucose. So can I say that this particular transporter is insulin dependent because insulin came and attached, and that's when the transporter went up ahead into the cell membrane and hence it is letting in some amount of glucose? Yes. Can I say it is insulin dependent? Yes. Now you are asked which one among them are actually insulin dependent. GLUT4, GLUT1, GLUT2, GLUT3. So about GLUT1, 2 and 3, none of in your NCERT biomolecules chapter it has not been mentioned. Only one thing is being mentioned and that is GLUT4. It is just said that GLUT4 basically is going to allow the transport of glucose. So please understand, it is the one that allows the transport of glucose within the cell, yes, definitely, and it is also insulin dependent. Only if insulin comes and binds, that's when this all thing would be happening. It is highly dependent on insulin. Will you ever forget it? I hope you not. Okay, so this is your answer for the sweetest question out there. Okay, next, which of the following have carbohydrate as a prosthetic group, as an additional group which is there? Okay, glycoprotein, chromoprotein, lipoprotein, nucleoprotein. Carbohydrate is usually recognized by glyco word. Yeah, glycogen, glucose, all them, then glycoprotein, glycolipids. Glyco term signifies what? Carbohydrate. So over here you are asked that which of the following have carbohydrate as a prosthetic group? It would be glycoprotein. Lipoprotein means lipid and protein is attached. Okay, lipid is the prosthetic group. Nucleoprotein means nucleic acid is attached to the protein. So nucleic acid is basically the prosthetic group. Chromoprotein means some pigment is there which is attached to the protein. That is the prosthetic group. Over here your answer would be glycoprotein because you have to look for the carbohydrate which is up there. Okay, cool. Next, which one of the following statement is wrong? Uracil is a pyrimidine, glycine is a sulfur containing amino acid, sucrose is a disaccharide, cellulose is a polysaccharide. Which statement is wrong? Uracil is a pyrimidine, definitely. Okay, I told you in nitrogen and bases you have two type of things. One is purines, another one is pyrimidine. In purines you have A and G, my dear children, adenine and guanine. In pyrimidine you have T and C. Apart from T, in the case of RNA, it has uracil. It has uracil, my dear children. Okay, always remember that. Now, uracil is a pyrimidine, that is correct. Glycine is a sulfur containing amino acid. Glycine, I just told you, does it contain a sulfur? No. Basically, talking about glycine, it will be having an amino group, it will be having a carboxyl group, an H group, and also instead of in the R also, it has H group itself. It's the simplest one. So, glycine as a sulfur containing amino acid would be very wrong. Okay, now sucrose is disaccharide. Yes, sucrose is actually a disaccharide. Right, now it is made up of two sugars, right? Now cellulose is a polysaccharide. It is made up of multiple chains of what? Glucose molecule, my dear children. So many glucose comes together to form cellulose. So it is a polysaccharide, that is also correct. So what is wrong? Your B is wrong. Cool. let's go ahead. So this is what I was talking about. There's only H. Now, the chitinous exoskeleton of arthropods is formed by the polymerization of. Do not get a heart attack seeing this question. I know that certain things are not there in your NCERT, still it is being asked, but yes, you have to understand it, you have to remember it, yeah. So, chitinous exoskeleton. Chitin is made up of what? Chitin, which is a part of the exoskeleton of arthropods, it is made up of what? N-acetyl glucosamine, lipoglycans, keratin sulfate and chondroitin sulfate, or D-glucosamine. What is the answer? What is the chitin molecule made up of? It is made up of, it's not a molecule, chitin is basically a compound that is there, and it is basically made up of polymerization or monomer unit of N-acetyl glucosamine. This becomes your answer. N-acetyl. So, chitin is a long chain polymer of N-acetyl glucosamine, a derivative of what? Glucose. It is a derivative of glucose. Will you forget this? I hope you do not. You have to remember it. Cool? Now, going ahead. Carbohydrates are commonly found as starch in plants, plant storage organs. Which of the following 5 properties of starch make it useful as a storage material? You're not asked what is true for starch. You are asked what is that something that makes starch the perfect storage material? That it is easily translocated? Is it easily translocated? Chemically non-reactive? Yes, it is chemically non-reactive, which is why it is used as a storage. Storage things should not be reactive, right? So, this is one very important property. Easily digested by animals? Well, it is. But does it count for being a storage material? No. Osmotically, it is inactive. That means, for example, if I add up starch up there, okay, it won't, the other cells would not be losing up water. That it means. That is what it means. It does not, let's say, disturb the osmotic balance in there. Yes, starch, when it is there, it would not allow the disturbance of the osmotic balance which is up there. So, I say it is osmotically inactive. In the case of starch, if you put just glucose, what will happen? All the water will be coming in, and this and that, lot of things would be happening. There will be disturbance caused there. So, yes, starch is osmotically inactive, which is why it is considered to be a good storage substance. Now, synthesized during photosynthesis, synthesized during photosynthesis? Yes, it is. So, it is actually glucose is the one that is synthesized, but glucose is stored in the form of starch. But again, do you think it is a reason? No. What is the reason for this? The reason is 2 and 4. So, your answer becomes 2 and 4. Okay, because it is non-reactive and also it is osmotically inactive. That is the reason. Now, which of the following is a reducing sugar? Reducing sugar means this sugars, whatever thing is there, would be having a free end, okay, would be having a free end to donate, to accept, and a lot of other things, okay. So, galactose, which is just a sugar molecule, obviously it is not attached to anyone else, which is why it would be what? It would be a reducing sugar. It can undergo reactions like that. Glucanic acid, no. Beta-methyl galactosidase, no, because all of them have some groups attached to it. Sucrose is a disaccharide. Sucrose is a disaccharide made up of 2 sugars, which is why it won't be having what say a reducing end right over there. There is no free arm up there. Okay, because both of them are attached together. There is only one thing, and that is galactose. So, over here I have the reason why others are not. Gluconic acid is an oxidized form of glucose. The aldehyde group of glucose has been oxidized to a carboxylic acidic group, which is why it is now no longer available to be acting as a reducing sugar. Now, beta-methyl galactosidase is a glycoside. Okay, in glycoside, the anomeric carbon, the carbon which is a part of an aldehyde or ketone group, okay, is linked to another molecule, that is a methyl group, through a glycosidic bond. This linkage prevents the sugar ring from opening up to form a free aldehyde or ketone, making it non-reducing sugar. Same thing goes for sucrose. Sucrose is a disaccharide composed of glucose and fructose linked together, so you do not have a free end right there for making it a reducing sugar. So, your only answer is galactose, because it is what? Just a single unit of sugar which is up there. Okay. Now, cellulose, the most important constituent of plant cell wall, is made up of what? Branch chain of glucose molecules linked by beta-1,4 glycosidic bond in straight chain and alpha-1,6 glycosidic bond at the site of branching. First of all, is it branched? No, my dear children, it's linear. Cellulose is linear and it is made up of many glucose, and these glucose are linked via 1,4 glycosidic bond. What 1,4? Beta-1,4 glycosidic bond. Always remember that. I have a picture, I think. Yeah. When do you call it beta? Basically when the joining is happening in opposite direction. If both the OH group were right over here, my dear children, if both the OH group were right over here in the same line, you would call it alpha 1,4 bond. But over here it's there and one is over here. Okay, which is why you call this beta 1,4 glycosidic bond right over here. So it is unbranched, please remember that it is unbranched chain of glucose molecules linked together by beta 1,4 glycosidic bond. Branch chain of glucose molecule linked by alpha 1,6 glycosidic bond, no. Unbranched chain of glucose molecule linked by alpha 1,4 glycosidic bond, no. Your only answer this is this. Cool? Cool. Now, nucleotides are building blocks of nucleic acid. Each nucleotide is a composite molecule formed by nucleotide. I already told you there will be a sugar. On the first carbon of the sugar, what is attached? The nitrogenous base. On the fifth carbon of the sugar, this is the first carbon, this is the fifth carbon. Onto it, what will be attached? A phosphate group will be attached. Okay, so nucleotides are building blocks of nucleic acid. Each nucleotide is a composite molecule formed by base, sugar and phosphate. Okay, base, sugar and OH, base, sugar, phosphate, bracket n times, okay, and sugar and phosphate. So nucleotide, each nucleotide is a composite molecule formed of. You're asked about each nucleotide. You're not asked what is DNA or RNA, a nucleic acid made up of. In that case, you could have been put at the bracket over here and put N. But you're asked about just a single unit of nucleotide. A single unit of nucleotide is made up of base, sugar and phosphate. This becomes the answer. Okay. Cool. Next, which of the following nucleotide sequences contains 4 pyrimidine bases? 4 pyrimidine bases, my dear children. Pyrimidines are what? I told you it can be thymine, it can be cytosine, or it can be uracil as well. It can be uracil. So you need 4 of them. 4. So over here if you see there is GAT. This is 1. Okay. This is 2. This is 3 and this is 4. I got one which has 4 pyrimidines. I'll see this. So here it is 1. This is 2, this is 3. I just have 3 over here. If we talk about this again, 1, 2, 3. So what is your answer? This is obviously not the answer. Your answer would be this one, which has 4 pyrimidines. Okay, now if you're not knowing pyrimidines, you might feel, oh, what an easy question is this. But if you do not remember pyrimidines, what will happen? So always remember, you can remember like this. AG, OG, we say like that. No, we do not say like that, but still, okay. I'm being too dramatic, but that's how we remember. So AG which is there, it is what? Purines. So whatever is left behind would be what? Pyrimidines. Okay, okay. Next. Question number 18. A nucleotide is formed of, again, same question in a different manner. Purine, pyrimidine and phosphate. Purine, sugar and phosphate. Nitrogen based sugar and phosphate. Pyrimidine, sugar and phosphate. A nucleotide can be purine, it can be pyrimidine also. So over here nitrogenous base is written, that would be more easy. Or what say, that is more inclusive of all the things, both pyrimidines and purines. So I'll say this is your answer. Nitrogen base, sugar and phosphate. So nucleotide is formed of all these 3 things that you see over here. Cool? Cool, ma'am. Next. Ramachandran plot is used to confirm the structure of what? RNA, proteins, triglycerides or DNA. So Ramachandran plot is basically used to describe about amino acids, and what is that something that amino acids comes together and form? It is what? Proteins. So can I say it is used to confirm the structure of proteins? Will I be wrong if I say that? No, your answer is this. Ramachandran plot is actually used to confirm the structure of proteins, my dear children. Not RNA, not triglycerides, not even DNA. Okay? Okay. Next. Identify the substances having glycosidic bond and peptide bond respectively in their structure. Okay? Glycosidic bond and what? And you need peptide bond also. So chitin is okay, it will be having what? Glycosidic bond. It is made up of polysaccharide. Talking about cholesterol, no, it cannot be considered. Okay, it cannot be considered to be a sugar or a protein, so which is why it will be wrong. Glycerol is just a unit, is an alcohol molecule. Okay, would not be considering it to have a glycosidic bond. Trypsin is a protein, it's an enzyme, so it will be having a peptide bond, but it does not have the glycosidic bond, this guy. Cellulose, again it will be having the glycosidic bond. Lecithin, which is there, I told you it's a lipid. It's what? A lipid, and it's a part of your cell membrane. So again, no, lipid is not considered over here. You need peptide bond are there in proteins. So inulin, which is there, it is what? A carbohydrate, my dear children. It's a polysaccharide. It is polysaccharide made up of fructose. Fructose comes together to form inulin, always remember. Now insulin, which is there, it is a peptide hormone. Okay, it is a peptide hormone. So this is a polysaccharide, that is a carbohydrate. Carbohydrate will be having glycosidic bond between them, and this is what? A peptide, a protein. Okay, actually a peptide hormone. So it will be having what? Peptide bond between them. So it is made up of amino acids, which is why it will be having peptide bond between them. So this is your answer. This came in NEET 2020, very recent. Okay, so over here I have all the thing. Chitin, cholesterol. Chitin is polysaccharide with glycosidic bonds, but cholesterol is a lipid, a steroid structure, and does not contain peptide bonds. Glycerol and trypsin. Glycerol is a simple alcohol and does not contain glycosidic or peptide bond. Trypsin is a protein containing peptide bonds but not glycosidic bonds. Cellulose and lecithin. Cellulose is what? A polysaccharide with glycosidic bonds. Lecithin is a phospholipid containing ester bonds. Okay, so lipids have ester bonds. Always remember that. So lipids, whatever you see lipids, they have ester bonds. Proteins have peptide bonds, and what is called sugar has glycosidic bonds. Cool? Cool, ma'am. Now, which of the following biomolecules does have a phosphodiester bond? Phosphodiester bond. Where do you find phosphodiester bond? Amino acids in a polypeptide. This is peptide bond. Nucleic acid and nucleotide, I'll keep it for the end. Fatty acids in triglyceride, ester bond. Monosaccharides in a polysaccharide, glycosidic bond. Okay, monosaccharide and polysaccharides, and you're talking about carbohydrates. Fatty acid and triglyceride, again as I told you, ester bonds will be there. Nucleic acids in a nucleotide, my dear children. Okay, wait. Nucleic acids in a nucleotide. That's a wrong statement. It should be nucleotide in the nucleic acids. It would be more correct. Okay, I think there's a typo error. Yes, it should be like that. Nucleotide in what? In the nucleic acids. In that case, it would be correct. What is the answer for this then, my dear children? So, over here, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, you have the phosphate group. Okay, so this carbon and the phosphate group, which is known as, it is known as phosphoester bond. Now the same phosphate is also going to bond with what? 1, 2, 3, the third carbon up over here. So this is also a phosphoester bond, this is also a phosphoester bond. Together, can I call it phosphodiester bond? Yes. So basically, can I say nucleotide? This is a nucleotide, this is a nucleotide. Nucleotide in nucleic acid. Okay, nucleotide in a nucleic acid forms what? Phosphodiester bond. In that case, it would be correct. So it should be like this, and it should be like that. Okay. Okay. So please correct it. Okay, I think the question itself, there was a typo error probably. Yeah. Now, a segment of double-stranded DNA has 120 adenine. Okay, double-stranded DNA is there. There is 120 adenine and 120 cytosine bases. The total number of nucleotides present in the segment is, so in the segment there is what? Adenine is how much? 120. That means thymine will also be there, they will also be, because adenine pairs with thymine. I hope you know that. A goes with T and G goes with C, triple bond, right? And now cytosine, which is there, it is given 120. So over here guanine will also be 120. You are asked about how many, the total number of nucleotides present in the segment will be. So 120 into 4, my dear children, what your answer will be? 120 into 4 is what? 480. So hence your answer would be 480. A segment of double-stranded DNA. If it is mentioned single-stranded DNA, then you cannot say. A single-stranded DNA can have adenine, can have how many number of adenine and guanine and cytosine and thymine, we never know. Even if it's given adenine this much, tell the amount of thymine, I do not know. I can only tell if it base pairs with it. I'm talking about single-strand. In single-strand, there is no base pairing. So how can I say? There can be many more number of thymine also. Get it? So here it is double-stranded, which is why your answer would be 480. Going ahead, which is not consistent with double helical structure of DNA? A is equals to 2, C is equals to G. Correct. That is correct. Yeah. How many number of A in double stranded, right? It will be consistent. So A would be equal to T and G would be equal to C and vice versa. Correct. Density of DNA decreases on heating. Density of DNA decreases on heating. Yes, it does. It would be getting decreased on heating. It will be moving around, moving away from each other, okay, and the volume would be more if you consider like that. If the volume would be more and density is what, mass divided by volume, in that case it would be, if volume is more, the density would be less. So I will say the density of DNA decreases on heating. That will be correct. Now A plus T and C plus G is not a constant. Wrong. It is actually a constant, and that is what Erwin Shergav's rules say. Erwin Shergav had two rules. According to this, one rule was A plus T and just a second, okay, A plus G and C plus T is equals to 1. Is equals to 1. Will it be correct? Yes, it will be definitely correct. How many number of A will be there? That many number of T will be there. How many number of G will be there? That many number of C will be there, which is why it is equals to 1. Apart from it, he put down one more rule. He said that A plus T, okay, and C plus G is going to be constant for a particular species. For each species, it will be constant. Okay, the number of this and the number of this would be constant for a species. In different species, this number is going to change. So this is not constant would be a wrong statement to put down over here. Both A and B is wrong. So which is not consistent with double helical structure of DNA? This one. It is not consistent. Okay. Ayyo, where did it go? Come back again. It's not coming. Usually it comes like that. Okay, chalo. Next, match the following. Inhibitor of catalytic activity possesses peptide bond, cell wall material, secondary metabolite. So, what is the easiest? This one is the easiest. Cell wall material, chitin and fungi. In fungi, the cell wall is made up of chitin. In bacteria, it is made up of peptidoglycan. In the case of plants, it is made up of cellulose, right? Now, cell wall material is chitin in fungi. So, C goes with 3, C goes with 3. I just have one option, and with that one option, I got my answer as well. Now, let's see. A goes with 2. A is what? Inhibitor of catalytic activity. That is malonate. Yes, malonate actually is a what? Competitive inhibitor. Okay, it is an inhibitor of what? Succinate dehydrogenase. Is an enzyme that converts succinate into fumarate. Okay, and basically over here malonate is going to be acting like a inhibitor. So that is also correct. Now B goes with 4. Possesses peptide bonds. That means whatever you're talking about should be a protein. Yes, collagen is a protein, so I'll say it will be having peptide bond. Secondary metabolite. What is left? What are we left with? Ricin. And ricin is what? A toxin. Ricin is a secondary metabolite and it is a toxin. Okay, so your answer would become A. Okay, cool. Next. Consider the following statements. Coenzymes or metal ion that is tightly bound to enzyme protein is called prosthetic group. Listen to me carefully. So for enzymes, when you want the enzyme to work very proficiently or something like that, you need the help of cofactors. You need certain cofactors, and you have 3 types of cofactors. One is prosthetic group, which is a non-protein group which is there, and it is actually very firmly attached. Okay, it is very firmly attached to the enzyme which is there. And now then after that you have coenzyme. Coenzyme is also non-protein, and basically they are transient. Okay, they attach for some time and they go off. Then you have metal ions, which again is going to help in increasing the proficiency. Now coenzyme or metal ion that is tightly bound to enzyme protein is called prosthetic group. Coenzyme and metal ion that is bound to enzyme protein is a prosthetic group. No, prosthetic group is a different thing, a different type of what cofactor, my dear. All these 3 things are different type of cofactor. So coenzyme or metal ion that is tightly bound to enzyme protein is prosthetic group would be a wrong statement to say. Prosthetic group itself is something else. Now a complete catalytic active enzyme with its bound prosthetic group is called apoenzyme. There is an active enzyme and then you have the prosthetic group also. This is known as apoenzyme. Wrong. Apoenzyme is the actual enzyme, proteinaceous enzyme which is there. That is the apoenzyme. Onto which if your cofactor attached, it will be something else. That is known as holoenzyme. Together it will be known as holoenzyme. So my dear children, the enzyme protein which is there, it is known as apoenzyme. Apoenzyme plus cofactor plus any cofactor which is there, it will be forming something that is known as hollow enzyme. It will be known as hollow, as in whole, hollow enzyme. So can I say both the statement would be wrong? Yes, both A and B are what? False. Cool? Yes. Going ahead, come on, this time you have to come over here. Yes. So a prosthetic group is a non-protein chemical group that is tightly covalently bound to an enzyme or the protein. Or the coenzyme is also a non-protein organic molecule that helps an enzyme catalyze the reaction. However, coenzymes are often loosely or transiently bound to the enzyme. Metal ion can also act as a cofactor for enzyme. Done? I told you. Now, which of the following statement is correct with reference to enzyme? Now, you have to tell me. Hollow enzyme is equals to apoenzyme plus coenzyme. Hollow enzyme is equals to apoenzyme, that means the natural enzyme which is there, plus coenzyme. Coenzyme is what? Basically cofactor, a type of cofactor. So, chalo, this is correct. But let's see if we have just cofactor also, just like this. Hollow enzyme is equals to apoenzyme plus cofactor. If it is mentioned like that, it will be more correct. Now, coenzyme is equals to apoenzyme plus hollow enzyme, wrong. Hollow enzyme is equals to coenzyme plus cofactor. Coenzyme is a type of cofactor. So, why do you have to put it separately? And where is apoenzyme? No. And this is also no. So, your answer becomes this. This is the most appropriate option that we have over here. Hollow enzyme is equals to apoenzyme plus coenzyme, which is a type of cofactor. Next, a non-proteinaceous enzyme. That means it is not made up of protein, but it is still an enzyme. What is it? Lysozyme, ligase, deoxyribonuclease, ribozyme. What is the answer? Your answer is ribozyme. I hope you remember 23 sRNA. Okay, remember my dear children, it's there in the case of prokaryotes. They are going to act like, okay, they are basically RNAs, yeah, and they are going to act like what? Enzymes, which is why we say RNA basically is acting like a genetic material and also an enzyme. So ribozyme is a non-proteinaceous enzyme which is there, and that's all. Please remember it. It helps in the formation of peptide bonds. You know, ribozyme is the one that helps in the formation of proteins. Ribosome goes and reads what the mRNA, translates it, and while translating it, it is translating into protein, right? So, this amino acid which comes together has to bond together, form peptide bonds. Who does it? Okay, so please remember that. Now, which of the following describes the given graph correctly? Let's see. Endothermic reaction with energy A. Okay, this is the graph. Please go through the graph once. Potential energy, reaction is there, substrate is there, product is over here. This is something. Okay, now it is said that it is endothermic reaction. First of all, is it endothermic reaction? Substrate is over here, product is over here. Is it endothermic? That means, is it taking in energy? In that case, it should be going up, right? No, but it is losing energy, which is why it will be what? Less. Product is here. The product will be here, my dear children. Potential energy is very less. Okay, so it is not endothermic. It will be exothermic. It has lost the energy. So wherever you see endothermic, you give a cut to that. Okay, now going ahead to the next thing. Exothermic reaction with energy A in the presence of enzyme, and this in the absence of enzyme. So what is this representing? This is representing something that is known as activation energy. Activation energy, okay, that is basically for a substrate to go to the transient state. Okay, that amount of energy is known as activation energy. So if enzyme is not there, it will need higher energy for going into that state. If enzyme is there, it will be quick. So activation energy will be what? Less, my dear children. So can I say this is where the enzyme is there and this is where the enzyme is not there? Okay, so A represents where the enzyme is there and this represents the absence of enzyme. So exothermic reaction with energy A in the presence of enzyme and B in the absence of enzyme. Okay, exothermic reaction with energy A in the absence of enzyme and B in the presence of enzyme. Wrong. Your only answer would be this. Exothermic reaction with energy A in the presence of enzyme and B in the absence of enzyme. Okay. Yes. Now, select the option which is not correct with respect to enzyme action. Let's see. Substrate binds with enzyme at its active site, which is not correct. Substrate binds with the enzyme at its active site, that is correct. Addition of a lot of succinate does not reverse the inhibition of succinic dehydrogenase by malonate. I told you there is an enzyme succinic dehydrogenase, and basically this converts succinate into fumarate. Yeah, but now there is another substance, that is malonate, that will be coming and competitively inhibiting. How does it competitively inhibit? Basically it fights with the actual substrate, that is succinate, and says that, hey, you cannot bind, I have to go and bind, I will bind. Okay, so basically that is happening. It is said over here that addition of a lot of succinate does not reverse the inhibition. So that means even if you add a lot of succinate, still malonate is the one that will go and bind. Do you think that's true? It is incorrect. Addition of lot of succinate actually reverses the inhibition of succinic dehydrogenase by malonate. This is wrong because it does reverse. Here it is written, it does not reverse. Now a non-competitive inhibitor binds the enzyme at a site distinct from that which binds the substrate. Okay, so for example over here you have this site where it binds with the substrate. A non-competitive inhibitor will come and bind somewhere here and change the entire structure and basically does not allow this substrate to bind over here. That is how non-competitive inhibitor works. If it is a competitive inhibitor, it will be having the same structure, it will try to come and bind over here, competing the actual one. So this one is a Dracula, this one is a normal one. So this is malonate, this is succinate, and this is what? Succinase dehydrogenase, if you're talking about this particular thing. So if you have lots of succinate, my dear children, obviously this guy would be losing up. Okay, but over here it is said that it does not make any sense. So that is why it is wrong. A non-competitive inhibitor, I told you, it will come and bind it elsewhere and change the whole structure, which is why the substrate would not be able to come and bind. Now malonate is a competitive inhibitor of succinic dehydrogenase. Yes, it is. So what is your answer? Which is wrong? This B is wrong. Cool? Okay. Transition state structure of the substrate formed during an enzymatic reaction is, I told you, there is a there is a substrate to becoming a product, there is a state in between, that is known as transition state. And this transition state structure, my dear children, what do you call? Transient. It is transient and unstable. Transient as in it is not forever there, it is going to what? Change to something else. So that is why it is known as transient, and my dear children, it is totally unstable as well, which is why it converts into what? The product. If it's stable, probably it won't be converting into the product. So it is transient and unstable. Transition state structure of the substrate formed during an enzymatic reaction is transient and unstable. Not permanent and stable, not transient but stable, not permanent but unstable, wrong. Just transient and unstable. That is the correct answer. Going ahead. A competitive inhibitor of succinic dehydrogenase is. A competitive inhibitor of succinase dehydrogenase is what? Just told you so many times. Alpha ketoglutarate, malate, malonate, oxaloacetate. Your answer is malonate. This is the competitive inhibitor of succinic dehydrogenase. So many questions have come around this particular topic, so make sure you know this. Next, an organic substance bound to an enzyme and essential for its activity is called an organic substance. Organic. Okay. Bound to an enzyme and essential for its activity is known as what? Isoenzyme, coenzyme, holoenzyme, apoenzyme. Apoenzyme is basically the protein enzyme. Holoenzyme is basically apoenzyme plus some cofactor. Isoenzyme is something else. What are we left with? Coenzyme. So coenzyme is that, I told you, it's not protein. It's not made up of protein. Coenzyme is an organic substance that was bound transiently for some time. Okay, for some time, and that is really essential for the working of the enzyme. So your answer will be B. Now, 33rd. Which one of the following statements regarding the enzyme inhibition is correct? Competitive inhibition is seen when a substrate competes with an enzyme for the binding to an inhibitor protein. Wait, you have to find what is correct. So competitive inhibition is seen when a substrate competes with an enzyme. Why does the substrate compete with an enzyme? Substrate will, basically inhibitor will compete with the substrate, right? This is wrong. Competitive inhibition is seen when a substrate and the inhibitor compete for the active site on the enzyme. Yes, the substrate and the inhibitor, okay, they are competing for what? For getting into the active site of the enzyme. That would be correct. Non-competitive inhibition of an enzyme can be overcome by adding large amount of substrate. Non-competitive inhibition, my dear, I already told you. Basically, suppose this is how it was initially. A non-competitive inhibitor came and attached over here at this particular point. What happened? Okay, this was the substrate. Now, because this guy is attaching over here, it is not competing with that. It is attaching over here and now it's going to change something up over here. It's going to change the structure probably. So it looks like, over here, do you think it can come and attach? No. So non-competitive inhibition of an enzyme can be overcome by adding large number of, whatever large number of substrate you add, do you think it can come and fit over here? No, my dear children. This is because the whole structure has been changed. So this is wrong. Non-competitive inhibitors often bind to the enzyme irreversibly. Wrong. It is reversible. Okay. So what is the answer? Competitive inhibition is seen when the substrate and the inhibitor compete for the active site on the enzyme. This is the only thing that is correct. Next, 34. Enzymes, vitamins and hormones can be classified into a single category of biological chemicals because all of this. Enzymes, vitamins and hormones can be classified into a single category of biological chemicals because all of this what? Helps in regulating metabolism, are exclusively synthesized in the body of a living organism, are conjugated proteins, enhance oxidative metabolism. What is the answer for this question? Enzymes, vitamins and hormones can be classified into a single category because all of them are going to help in regulating what? Something that is known as metabolism, which is why your answer would be A, nothing else. Okay, it helps in regulating metabolism. Now, in which one of the following enzymes is copper necessarily associated as an activator? Carbonic anhydrase, tryptophanase, lactic dehydrogenase and tyrosinase. In which one of them do you think copper is necessarily associated as an activator? So, in the case of carbonic anhydrase, you need something and that is zinc. Remember that, okay, remember that at least. So, over here the answer is tyrosinase. This is the one where you need copper, something that is not there in your NCERT, I do agree, but yeah, this is the answer. So, tyrosinase is a copper containing enzyme that catalyzes the first step in the melanin biosynthesis. You know, melanin is being produced with the help of tyrosine. So, tyrosine has to convert into something and this something has to convert into melanin. So, that's how things works. Okay. So, it is involved in the oxidation of tyrosine to DOPA quinone and subsequently in the production of melanin pigments in the skin, hair and eyes. Copper ions are essential cofactors for this particular activity. Carbonic anhydrase uses zinc as a cofactor. Okay. Tryptophanase requires pyridoxal phosphate, a derivative of what? Vitamin B6, right? And lactic dehydrogenase uses NAD plus as a coenzyme. So, all of them have different. If we talk about tyrosinase, they need copper. Okay. Role of an enzyme in reaction is to what? When enzyme is there, what does it do? Decrease the activation energy, increase the activation energy, inorganic catalyst, none of the above. It is going to decrease the activation energy. Little amount of activation energy is needed. First, okay, the substrate would be forming the transient state and then the product. So, that fast thing should be there. That is why we say we use enzyme, because we want the process to be really fast. So, can I say it decreases the activation energy? Yes, my dear children. Role of an enzyme in the reaction is to what? Decrease the activation energy, not to increase it. So, which is why your answer would become A. Okay. Now, cofactor prosthetic group. Remember, you said something about it. Prosthetic group is a part of holoenzyme. It is what? So, cofactors are of three types: prosthetic group, coenzymes and then metallic ions, right? So, over here I asked about prosthetic groups. They are the ones that are firmly bound, okay? They are firmly bound to what? The enzymes. Always remember. Coenzymes, loosely bound, transiently bound. Okay, metal ions, they have not mentioned, but anyways, it is loosely attached to organic part, wrong. It is loosely attached inorganic part, wrong. Accessory non-protein substance attached firmly, yes. It is not made up of protein, that is why it is written as non-protein substance attached firmly. It is correct. None of this would be wrong. So, what is the correct answer? Your answer is this: accessory non-protein substance attached firmly. So, cofactor is a part of holoenzyme and this is the thing. Cool? Yes. Next. Enzymes having slightly different molecular structure but performing identical activity are. So, you have been seeing this option for quite long. I was not telling the answer because I wanted you to answer this. Enzymes having slightly different molecular structure but performing identical activity are what? Holoenzymes? No, holoenzyme means apoenzyme plus cofactor. Apoenzyme, so you know, is just the protein part of the enzyme. Coenzyme are the cofactors. Isoenzymes are the one, that is your answer. Enzymes having slightly different molecular structure but performing identical activity is isoenzyme. Okay, cool. Next, The figure given below shows the conversion of a substrate into product by an enzyme. Okay. In which one of the four options the components of reaction labeled as A, B, C, D are identified correctly? So over here your A is there, your B is here, your C is here, your D is here. You have to tell what is what. So we just know that B is what? Potential energy which is there. B is potential energy. So I can see one option like that. I can see one option, my dear children. If you're not able to see, I'll zoom it and show it to you. Wait just a second. Yeah. So there is only one option where B is potential energy. See, this was a graph. So this is B is the potential energy. This is activation energy with the enzyme. This is activation energy without the enzyme. What is A, my dear? Transition state that we are having. So B is potential energy. There is only one option like that. Hence this becomes your answer. So B is transition. Okay, wait. A is transition state. B is potential energy. C is activation energy without the enzyme, and D is activation energy with the enzyme. So hence your answer becomes this. Cool? Cool, ma'am. Now, last question. The curve given below shows enzymatic activity in relation to three conditions: pH, temperature and substrate concentration. What do the axes represent? You have y-axis and you have x-axis over here. What do they represent? My dear children, y-axis represents what? The enzymatic activity. Okay, x-axis can be either pH or temperature. Why I am telling you is that, yes, when the pH and the temperature is kind of high at this point of time, slowly it will obviously enzyme activity increases, but after a certain point of time, okay, after the optimum pH or temperature, it will be dropping down. If you are increasing the temperature more, the activity is not going to be great. It is going to be less. Okay, it is going to be bad. You get it? So please remember, optimum pH is the pH where it works the highest. Anything less than it is going to mean that the enzymatic activity is going to be lesser. That's all. Which is why the graph is like this. Okay, so there's the optimum pH, that's all. Then there's the optimum temperature as well. So basically y-axis is enzymatic activity. I have two options like that. Over here one is temperature, other is substrate concentration. For substrate concentration you have another graph. For temperature and pH you have a graph like this, which is why this becomes your answer. B would be your answer for this question. Cool? I hope you got it. And with that, we complete all the 40 questions from biomolecules, my dear children. I hope you got it. Okay, if you have any doubt in any questions, do let me know. Okay, do let me know if you have any doubt. I am here to help you out with that as well. So that's all. And take care of yourself. Do not forget to subscribe to the channel if you have not subscribed yet. And please do let me know all whatever is your concern. You can let me know if it is in if it's something that I can do, I'll definitely do something about it. Yes. So bye bye children. Take care of yourself.